As we know, each myofibril is having alternate arrangement of actin and myosin filaments. We are going to see that actual arrangement working as a functional unit in the form of sarcomere. Now, pay attention that how the sarcomere is having the alternate arrangement and how does it function. That is studied under sliding filament theory. But for understanding that filament theory, you need to know what is the structure of sarcomere. Supposedly, we are inside a muscle fiber, all right. Inside a muscle fiber, this is the line of distinction that separates it from other muscle fiber. There would be arrangement of actin and myosin filaments in such a way so that it enables the movement of the muscles and it results in contraction. Not entirely the muscles, the movement of the protein filaments so that the contraction takes place and hence the relaxation also takes place. So, for understanding the sarcomere, first of all, there is a membranous line that is known as Z line and another Z line. So, the region between two Z lines would be termed as a sarcomere. First thing that you need to remember. Right in middle of Z line lies another membranous separation that is known as M line. Okay. There are two Z lines which separate, then there would be another Z line. This is the functional unit of the muscle, skeletal muscle, separated from the other unit by two Z lines. So, Z line is the separation point. Right in the middle lies a M line and to the Z line is attached actin filament that is light and isotropic, like this. Actin filaments are arranged alternately with myosin filaments, but for once we are seeing that in Z line what that arrangement is to the both sides of Z line. So, would be in the other case, these are actin filaments. Please remember, actin filaments lie towards the periphery of the myofilament. These itself is a myofilament, but this is a myofibril that we are discussing. These are actin molecules. Now, actin is a protein, you know. We discussed in the previous lesson that it is anisotropic, it is light, while the myosin is pardon me, I used anisotropic for actin. It is not anisotropic, it is isotropic. Myosin is anisotropic. These things have to be remembered. If you can associate these things with your knowledge of chemistry, then you can analyze it in a better way. These are the actin filaments which are joining to the Z line. That's why I made Z line in the beginning itself, so that you can associate that each filament is joining with the Z line, and because this joining is going to help entire sarcomere, that is the functional unit, to work as an entity for contraction and relaxation. That is why we have the filaments attached to these Z lines. So, I am making it darker so that you get an idea that this is a Z line. Okay, and in between there is another membranous line that is known as M line. Now, from this M line, what is important to note that there is stretching of myosin filaments to both the sides, just like actin was attached to the Z line. Alternate arrangement that we had discussed, yes, it is present. These are filaments, long filaments which are going to bring contraction and relaxation. Something is dark, something is light. That is known as sarcomere. Now, what is light, what is dark? Let us see that. This part, as you can see, pardon me, sorry. This part is myosin. Okay. Now, see this. Because we discussed that actin filaments are isotropic and they are light, so this particular part is known as that part which does not have myosin present, that part which is having only actin. See this, myosin would have come from the other side over here as well. So this particular zone where there is only I band present is known as I band or I zone. Okay, this is the I band. This happens to be the dark one. So this is A band. Okay. Now because this myosin is darker and it extends till over here, so the A band would be larger. Now this part where there are only myosin filaments and M line in between, that is known as H zone, and this describes the sarcomere, the functional unit of muscles. Now inside a muscle fiber there is present a sarcomere. This particular zone from between one Z line to the other Z line is known as a sarcomere, which has alternate A and I bands, and this sarcomere is responsible for the contraction of muscles. Sarcomere starting from one Z line which has actin fibers subtending inside, then there is an M line that is also membranous line that has myosin fibrils that are coming inside towards it. They are thick filaments. So we have a darker A band which appears darker of course due to overlapping, and a shorter I band which is present due to single actin filaments. A small I band, then there is an A band, then there is a small I band followed by a small I band followed by an A band. So this is what is present inside a sarcomere, and from here we are going to see the next topic that we deal with is the structure of both these proteins we are going to understand, and by understanding that structure we can decipher that how this contraction is brought about. Because what happens during sliding filament theory, I will give you an idea that these fibers of actin, they move inside, both the fibers they move inside. As they would be moving inside into the sarcomere or into the H zone, there would be contraction of Z line, and that contraction would bring about entire contraction because it is in connection with next one and next one, and you know that there are no gap junctions present inside the muscular tissue. So the contraction would move further and further due to the contraction of actin filaments getting inside the H zone. So this is all about sarcomere. We are going to understand it in a better way when we understand the sliding filament theory. For that you need to see the next lesson.